plasmodium in man. The human face. The human face. Then first the sporozoite is the sporo sporozoite is the infective stage to the man, right? In man, the plasmodium reproduces. Plasmodium reproduces. In man, the plasmodium reproduces by asexual reproduction. So this is called zygogony. What is this? Zygogony. The zygogony occurs in liver cells and RBC, right? So in liver cells it is called as hepatic zygogony, right? In RBC, in RBC, see, in RBC, erythrocytic zygogony, right? So now hepatic zygogony was discovered by the Short and Garnham named scientist. In that, the sporozoites are the infective stage to the man. Sporozoites are the infective stage to the man. Hepatic zygogony was discovered by the Short and Garnham, and the sporozoites are the infective stage to the when a mosquito bites a man. When a mosquito bites a man, nearly 2000 sporozoites released into the blood of the man through the saliva of the mosquito. So how many sporozoites enters into the body? 2000 sporozoites are released into the blood of the man through the saliva of the mosquito, right? So within half an hour, they reach the hepatocytes from the blood, where they start pre-erythrocytic and exoerythrocytic cycle. Nearly 2000 sporozoites are released into the blood of the man by the saliva of the mosquito. Within half an hour there is the liver. Liver is nothing but a hepatocyte from the blood, where they start the pre-erythrocytic cycle and exoerythrocytic cycle, right? So now we discuss about the pre-erythrocytic cycle, right? Whenever the mosquito bites, the sporozoites reach the liver cells and they transform into trophozoites. Whenever the sporozoite enters into the liver cell, they transform into trophozoites. Right? So these trophozoites feed on the contents of the hepatic cells. The trophozoites, so in perierythrocytic cycle, the trophozoite feed on the contents of the hepatocytes or hepatic cells and assume the spherical shape and grow in the maximum size. This stage is called as Shy-Giant stage. What is the stage? Shy-Giant stage. Shy-Giant stage, what happens here? Trophozoite grows in the maximum size and that feeds on the hepatocytes and becomes spherical shape. So this spherical shaped structure is called as a Shy-Giant stage, right? So the Shy-Giant stage, the nucleus divides several times mitotically, followed by the cytoplasmic divisions. So resulting the approximately 12,000 daughter individuals called cryptozoites are first generation merozoites are produced. The giant, the nucleus divided several times mitotically, followed by the cytoplasmic divisions. Giant, the nucleus divided several times mitotically, followed by the cytoplasmic divisions, and resulting in the approximately 12,000 daughter individuals. They are called as cryptozoites or first generation merozoites. Cryptozoites are first generation merozoites are produced in the pre-erythrocytic cycle. Pre-erythrocytic cycle, right? So next cycle is exoerythrocytic cycle. So in exoerythrocytic cycle, if the cryptozoites enter the fresh liver cell, they may be undergo similar to that of the pre-erythrocytic cycle produces second generation merozoites. So pre-erythrocytic cycle, first generation merozoites are produced, and exoerythrocytic cycle here, second generation merozoites are produced. What are produced here? Second generation merozoites are produced. The second generation merozoites are called second generation merozoites are called as metacrypto metacryptozoites. Metacryptozoites. Second generation merozoites are called as metacryptozoites. So these metacryptozoites are two types. How many types are there? Two types are there. One is the macro metacryptozoite and another one is the micro metacryptozoite. So the micro is smaller, macro is the larger. So this will take approximately 2 days. This will take approximately 2 days time, right? So macro metacryptozoite attack fresh liver cells. Macrometacryptozoites attack the fresh liver cells, and micrometacryptozoites attack the RBC cells. RBC cells. So when the micrometacryptozoite attack the RBC cells, they continue the erythrocytic cycle. Erythrocytic cycle is continued by the micrometacryptozoites. Pre-patent period. Pre-patent period. So what is the pre-patent period? Interval. The interval between the first entry of plasmodium, in the blood. First entry of the plasmodium in the blood in the form of sporozoites. So these sporozoites are first entered into the body, and second entry of the plasmodium, in the blood in the form of cryptozoites. What is that? Cryptozoites. This is called pre-patent period. What is pre-patent period? First interval between the first entry sporozoite, second entry cryptozoites. This is called as a pre-patent period, right? So what is the pre-patent period? Approximately 8 days, right? So during this time the parasite increases in the number, and host does not show any symptoms of the disease. It is only means of multiplication. Multiplication. The host does not show any symptoms. The only parasite only grows in the number, right? That is pre-patent. Next, erythrocytic cycle. This is observed, or this is first described by Camillo Golgi. That's why this is called as Golgi cycle. This cycle is initiated by cryptozoites or micrometa-cryptozoites. Cryptozoites are the first entry in the body and micrometa-cryptozoites is second entry body. So both can initiate, both can start the erythrocytic cycle, right? So pre-erythrocytic cycle and exoerythrocytic cycle produces the cryptozoites and micrometa-cryptozoites. So in this cycle in the RBC, these stages assume spherical shape and transform into tropho. In the RBC, these stages assumes spherical shape and and transform into trophozoite. Transform into trophozoite. Transform into trophozoite. These trophozoite, one vacuole, appears, which is gradually enlarges, and that pushing the cytoplasm, and and nucleus to the periphery. So nucleus to the periphery, right? So after that, now it looks like a finger ring. That looks like a finger finger ring. That looks like a finger ring. So this stage is called as signet ring stage. This stage is called as a signet ring stage, right? So it loses it loses the vacuole. Then then this develops a pseudopodia, and this develops pseudopodia and transform into amoeboid stage. So this amoeboid stage, what happens? This amoeboid stage feeds on the components or contents of RBC and increasing the size. So amoeboid stage feeds on the contents of the RBC and increasing the size. As a result of RBC grows almost all double, because the parasite is settled in the RBC. That parasite settles in the RBC. That's why that grows in the size. This process is called as hypertrophy. What is hypertrophy? Increasing the RBC size by basing on the development of the parasite. This is called as a hypertrophy, right? So this amoeboid stage, it digest the globin, and it digests the globin from the heme. హిమోగ్లోబిన్లో ఉండేటువంటి గ్లోబిన్ను డైజెస్ట్ చేస్తుంది, అండ్ ద సాల్యుబుల్ హీమ్ ఇంటూ the soluble heme into insoluble hemogyne that convert into insoluble hemogyne. So this hemogyne is called as malarial pigment. What is this pigment? Malarial pigment. During this stage, small red colored small red colored dots appears in the plasma. Dots appear in the cytoplasm of RBC. What is cytoplasm? Plasma. So the plasma contains some dot-like the structure. These are called as Scheffner dots. What is the Scheffner dots? Scheffner dots, these are believed to be the antigens released by the parasite. Antigens. These are antigens, right? So then of the plasmodium loses the pseudopodia and further increases the size, occupy the entire RBC and become giant. That's now the plasmodium loses the vacuole or that loses the pseudopodia and further increasing in the size and the entire RBC and this is known as giant. This stage is called as a giant. So the giant undergoes giant. So the giant undergoes the schizogony and produces 12 to 24 erythrocytic merocytes. They are arranged in the petals of rose flower like structures. How many are there? 12 to 24. So this stage is called as a rosette stage. This stage is called as a rosette stage. Finally, the cryptozoites bursts and releases the merocytes along with the hemogyne in the blood. This cycle completes approximately in the 48 hours. 48 hours.